Hi everyone, today you guys are going to do a virtual lab. So today you guys will be working with fruit flies, which are kind of the genetic whipping boys. So they're used in a lot of experiments because they're so easy to reproduce and to modify. But they're also sometimes a pain in the butt to work with because they're so small and it's really hard to keep them in control and dissect them, especially if you're new at it. But because we have great technology in this age, we can do our experiments virtually, online. So this is a long list of directions, but basically what you're going to do is you're going to go to this website, and this is where you're going to conduct all of your labs. And one important thing is to never back out. It's going to delete all of your work and you're going to have to start over. Okay, so these directions are pretty much straightforward. And before we go through them, I'm going to show you that there are 4 different crosses that you will work with today. So this is cross 1, where you have sepia. This is a color male with wild type of eye color, and wild type means it's just normal, no mutations. So this is the first cross you're going to look at. And your second cross is going to be sepia eye normal wing male with wild type, which is another word for normal eye or normal anything, and vestigial wing female. Your third cross is going to be wild type male, so no mutations in any of its features, and a white-eyed female. And your fourth cross is going to be something that you can design. Okay, so let me just sort of go into the website to show you how this can work. Okay, so this is the website that you guys are going to go into. And the first thing that you're going to do is order flies. So you're going to click on this computer to order flies. And once you get in the computer, you have these 3 different options. You can either order flies, analyze your results, or return to your lab. So because we first need to order these flies, we're going to click on this one. Okay, and these are the different traits that you can choose for any mutations. And here you can choose the gender. So remember, in our first cross, we have a sepia eye color male. So I'm going to click on male, and then we're going to go to eye color and click on sepia. And then we're going to click on this add to cart to buy it. And then for female, remember it is wild type. So we're not going to choose anything. And it says wild type here. So we're going to add this one to cart as well. So now we're going to go to our cart, check that these are the flies that we want. We want sepia-eyed male and wild type female, and you're going to click checkout. And it would be also good to add these flies to your notebook so you can keep it track. Okay, now our flies that we ordered have come. And now each of them have been put into this mating jar. So if you look at this, if you hover over it, you can see that we have a female wild type mated with a male sepia eyes. Now we're going to click on it to put it into the incubator, and it's going to allow the flies to mate. So usually it takes about 2 weeks, so that's kind of what we're waiting for. And if we click on this jar again, it's done, and we click on the incubator, click on this mating jar, and now they're knocking off or kind of putting the flies to sleep as we sort the different offsprings. So if we click sort flies, you have these flies now sorted, and if you hover over it, you can see that one group is female wild type and one group is male wild type. So we want to send this data to the computer lab, and according to the instructions, we want to do another cross. So we're going to the computer later, and we want to return to the lab. So the flies that we just looked at were our first generation. It was our F1. Now we need to create our F2 using the offsprings we just looked at. So, okay, actually, we have to go back to our sort. And then what we're going to do is if you click the female, you can use this option, use a new mating, and then zoom out, click the male, and also click use a new mating. Okay, and then you're going to zoom out. Now, if we return to lab, we have a new mating jar here that's waiting for us. So we're going to click on that, and they go into the incubator and start mating. So after 2 weeks, we can now look at, remember this is cross 1, this is cross 2, we can look at cross 2 and see what offsprings we have. So under the microscope, we're going to sort these flies. Remember this is F2. And now we have 4 different groups, and if you hover over them, you can see what they are, and you can also see the quantity here. So this is basically what you're going to be doing for all the crosses. And then here in your lab worksheet, you're going to create 2 data tables. One is going to be for your F1. So you're going to write the phenotypes and the number for each phenotype. And the second data table is for your F2. And you're going to write the phenotypes and numbers for each phenotype of F2. And then you're going to hypothesize what kind of inheritance pattern is it. Is it a recessive inheritance? Is it dominant? Is it X-linked? Is it codominant, incomplete dominant? Anything that we've learned. Okay? And then you're going to do this hypothesis justification and testing. So you're going to follow the instructions here, and you're going, at the end, you're going to justify your conclusion using chi-square analysis that we learned in the previous lesson. So you have your chi-square table here in page 2, and some directions for the analysis. So basically follow all of these steps in order, and do that for each cross, all 4 of them. Okay? And what you can also do for a chi-square analysis, if you go to, so we're going to send this data to the computer. If you go to the computer, you can analyze your results here using chi-square analysis. So if you click this, you can either go to cross 1 or cross 2. And if you click on this checkbox, you can either look at the sex or ignore the sex. And it's going to show you the number of flies and the proportion of each phenotype in total. And what you can do is click this chi-square analysis for this one. I think we can ignore sex because we don't see much of a difference between the sex. And here, let's hypothesize that this is a 3-to-1 ratio. And let's view our results. Before we view our results, we have to test this hypothesis. So what this does for you is it calculates the chi-squared value for you and the degree of freedom, and it calculates the level of significance for you. So if our chi-square test statistic is this number and our critical value is this number, we can see that our chi-square number is less than the critical value. So that means we accept our null hypothesis. So that's what you would be doing for your hypothesis justification. Okay. So I hope this is pretty much clear, and let me know if you have any questions either through email or through Zoom.